Hi, this is Miss Litton, and this is a tiny little team, and we are going to review the, are you out of here? We're just starting now. I told AP I would give them an additional review today.
Oh, aren't they like?
So we just did honors, now we're doing AP.
You were devoted.
Trying to be, you know. Have a good one, my friend.
All right, see ya.
Okay, so AP Bio Unit 2 essays, you have these posted where? On the anchor page. And I just want to read, that was, remember, my old organization, so now I've kind of compiled them right here for you. I know they're tiny, but you'll have access to them. So the first essay on here says, describe the model of a cell membrane of a eukaryotic cell and discuss different ways in which substances move across the membrane. In your discussion, be sure to indicate whether your examples are passive or active transport, right? So when you do that, you are going to definitely want, let me make this bigger, okay? You are going to want to be able to draw yourself a cell membrane, right? A simple one in case I ask you about this. What?
The ECM.
Yeah. Do you know everything I'm drawing right now? Okay, good. Okay, there's our simplified membrane. Okay, what would this be specifically called? Yes, but a glyco what? Protein. Oh, glyco, you were right. I'm sorry. Let me move this over here. Glycoprotein, and this would be called a what? Glycolipid. Good. Okay, because it's attached to a lipid. Okay, what is this right here? Cholesterol. And what is this right here? A protein. Good. And what is this right here? Phospholipid. So you would want to know what each of those things do. So a phospholipid is going to create a what? Barrier between the inside and the outside. Has like the fluidity of like olive oil. We know that this region right through here is hydrophobic. And we know that the outer part and the inner part are both hydrophilic. Okay? And so we know that if we have a protein spanning, an integral protein that spans the entire width of the cell membrane, then this interior part, it's got to be hydrophobic and the edges have to be hydrophilic. Okay? So we've got some other things in here as well. We've got cholesterol. Cholesterol helps us because if it's too cold, it's going to prevent us from freezing. And if it's too hot, it's going to hold the cell membrane together. So it plays that role. But you only see cholesterol in what kind of cells? Animal cells. Good. Glycolipid, glycoprotein are part of the glycocalyx, part of the extracellular matrix, cell-to-cell signaling, communication between cells, anchorage, holding cells together, cell recognition. It's all used during development as well. Proteins, we talked about several different roles for proteins. They could be used as what? Channels. They could be used as carriers. They could be used as enzymes. They could be used as what? Cell recognition. What else?
The receptors. They
could be used as receptors. So you want to make sure that you can talk about the different roles of each of those structures. Okay? So now I'm going back here. It says, describe the model. Let me make it a little bigger here. It says, describe the model of a cell membrane and discuss the different ways which substances move across. So now let's talk about how things can move across. So one way is to go through the what? Phospholipid bilayer. Okay, and when it goes through the phospholipid bilayer, it cannot be what or what? Large or charged. Okay, what's the second way it can move through? Okay, but I would use the protein next, but yes, that's a way. So if it's using a protein and it's going from whatever it is that you're moving, okay, if whatever you're moving is going from a higher concentration to a lower concentration, and so it's moving in this direction, okay, then the protein is, what do we call that? It's doing what?
Passive transport.
It's passive, and another word, facilitated. Because you're just, and you could do that if it was a what or a what? Channel or a carrier. But if you go the other direction, okay, if you go from a low concentration to a high concentration, that's when you refer to it as a what? Pump. Okay, because you're going against the concentration gradient. And the pump is going to require what? ATP. What's the most famous pump that we talk about all the time? The sodium-potassium pump. The third way to cross the membrane is through what? Endo and exocytosis. And if I do endocytosis and it's fluid, it's called what? Pinocytosis. And if it's solid, it is phagocytosis. And if it binds on, there's receptor-mediated endocytosis. Okay, perfect. So when you do endo and exocytosis, is that active or passive? Active, because it's going to require energy for that membrane to move. If it's going through the phospholipid bilayer, what's that going to be? Passive. Okay.
Unless it's going against.
Yeah. If it's going through the phospholipid bilayer, it's always going to be passive because you're not controlling that. Now, we talked about a specific channel that allows water to move. Do you remember what that's called? Aquaporin. Good. All right. Going back to small print. I think we did all that. Okay. The next question. A laboratory assistant prepared solutions of 0.7, 0.5, and 0.3 and 0.1 molar sucrose, but forgot to label them. So after realizing the error, blah, 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 design an experiment, okay, to figure out which one is 0.7, 0.5, 0.3, and 0.1. So tell me how you're going to perform it. What would be your flow chart? What would you expect? Okay, so let's talk about that. I have 7, 5, 3, 1. 7, 5, 3, and 1. Okay. So I have these 4 terrible beakers. Okay, and I don't know which one is which, but I know I have a, is it 1 or 0.1 molar? Do you remember?
I think it's 0.1.
Yeah.
No, it's 0.1. Okay, 1, 3, 5, 7. Okay, so I know one of them is 0.1, one of them is 0.3, one of them is 0.5, and one of them is 0.7 molar. Are we okay on that? Now, what am I going to do? What kind of experiment could I do in order to find out which is what? Go ahead.
You could add an enzyme.
An enzyme?
I'm thinking of a different experiment.
Okay, so let's try it.
You could put like a
Long
little
lab.
thing of dialysis tubing and just distilled water.
There we go. Did you hear that? Because it was perfect. Take yourself a beaker, fill it up with what? Distilled water. Right? What's the water potential of distilled water? 0. Right? Then you could get some dialysis tubing. And we know how to tie off that dialysis tubing, right? Using some string. Okay, then I could do this 4 different times, right? And I would put some mystery solution 1 in there for my first experiment, then mystery solution 2, mystery solution 3, and so forth, right? Now, in order to make this work, what would I have to do before I put it in the water?
You would have to wait.
I would have to weigh it. So I would have to get my initial mass, I'd take my final mass, right, minus my what? Initial mass, and put that over my initial mass. And that's the way I could get the percent change. And what would you predict would happen?
Lowest molarity would have the lowest percentage.
Yeah, the lowest molarity would have you would say, well, whichever one gained the least amount of weight, right? I would predict that that one would be, you know, percent change, because the dialysis tubing isn't all going to be exactly the same. I would predict that would be 0.1, and then whichever one changed the most, because water is always going to move from a higher water potential to the lowest water potential the most. So which up there has the lowest water potential of all?
The point one? Wait.
0.7, right? 0.7 would have the lowest water potential of all, and so it should gain the most mass. So I have to leave room for it to expand, etc. Now, what could I use as a control in this?
You should put another thing of distilled.
Another thing of distilled water in there as a basis of comparison. Okay, that takes care of essay number 2. So now let's look at essay number 3. Enzymes are biological catalysts. Relate the chemical structure of an enzyme to its specificity and catalytic ability. Then design an experiment to investigate the influence of pH or temperature on the activity of that enzyme and any information you could find out about the structure. Okay, so the first thing, let's deal with the first issue. It says, relate the chemical structure of an enzyme to its specificity. Why is it specific? And its catalytic ability. So let's deal with the first part, okay? What is the structure of an enzyme? What's it made out of?
An active site.
It's made out of what?
An active site.
Okay, you gotta tell me more. What is an enzyme made out of?
Oh, protein.
It's made out of a protein, okay? Now, we know proteins are built out of what? B-U-I-L-T. Built out of what? Amino acids. Do we know what primary, secondary, tertiary, and quaternary structure is? What is the primary structure? The sequence of amino acids. What is the secondary structure? Yeah, either an alpha helix or a beta pleated sheet. What's the tertiary structure?
It's the fold.
The folding of the secondary, right? And that is dependent on the what? R groups, good. And then what's the quaternary structure?
Multiple proteins.
Yeah, more than one chain. Good. So if I know the structure of an enzyme, okay, is built out of a protein, which has primary, secondary, tertiary, and quaternary shape, I'm relating this to its catalytic ability. Its catalytic ability is how well it can make reactions occur. Do you agree with that? Okay. So what's gonna affect how a reaction occurs if it has a functional what site? Active site. Now you can talk to me about the active site, and you could tell me that where the active site is, okay, is where it binds to the substrate. What determines the shape of that active site? This is this right here, right? What else can affect that active site? Yes, an inhibitor. What else can affect it, how well an enzyme works? pH. What else can affect it? Temperature. These are all things that are going to affect the structure, because temperature and pH can change. It's not going to, temperature and pH is not going to change the primary structure, but it could change the what? Secondary and the tertiary and the quaternary structure. Temperature and pH could alter those. If you alter that, you're going to alter the active site, which is going to affect the catalytic ability. We good so far? Sorry, no breaks since 7:20. Okay, I do have to pee. So then the B part says, design a quantitative experiment to investigate the influence of pH or temp on the activity of an enzyme. Is that what we did? Did we do an experiment where we affected pH or temp?
No.
No. How did we do it?
The amount of substrate.
The amount of substrate. Could you do the exact same experiment, but instead keep the concentration of the substrate the same, but change the temperature or the pH that the enzyme is at?
Yeah.
Probably the easiest thing to do would be to vary the temperature. Exactly. So you could do the exact same experiment you did before. Hydrogen peroxide's the substrate. Add in the catalase. Measure with the probe the amount of oxygen generated, because hydrogen peroxide breaks down into oxygen and water, and you would generate oxygen. You would be able to make a claim or a hypothesis, right? Our hypothesis could be, if I, what? Tell me how you would do this.
If I
Vary the temperature, then the amount of oxygen that is produced from the reaction will vary, right? Because enzymes work at a specific temperature, optimal temperature, right? So you could do it cold, you could do it room temperature, what would be body temperature, and you could make it really hot. Yes?
Let's say we go even further and could we hypothesize that, like, if we increase the temperature, wait, no.
Okay. Yeah. Go ahead, what was your question? Hi, howdy.
Hi, I have a question.
Okay, just so you know, you're getting recorded for YouTube.
Let's say.
Okay, all right. Okay, see ya. Go ahead.
How would you know that a null hypothesis would be more appropriate for the lab?
Oh, well, probably this would be one where you could do a null hypothesis, but I'm pretty sure I asked you because a null hypothesis is an easy out, and I'm sure in any experimental ones I am going to ask you to do a research hypothesis. But a null hypothesis is probably what I would actually go with. I would just say temperature does not affect the temperature of the enzyme does not affect the enzyme activity, and then I would measure it at different temperatures, and then I would use the chi-square test to evaluate it. But I'm probably going to push you and want you to, come up with a research hypothesis, I would guess. Okay? And then describe what information concerning the structure of an enzyme could be inferred from your experiment. Well, I would look and see if I have a really hot temperature and I get very little oxygen produced, probably the temperature denatured the protein, so its active site was impacted. Do you agree with that? Okay. All right, so that's that one. Okay, then this brings us, we only have two left. Okay. The next one says the net balanced equation for respiration is quite simple, but respiration is a complex process. Let me tell you right off the bat, you better be able to write the equation for me. You'd be surprised how many people can't. You ought to be able to tell me what gets reduced and what gets oxidized, right? And then it says, using diagrams and written explanations, describe the metabolism of glucose, including glycolysis, anaerobic fermentation, the Krebs cycle, and the electron transport chain. Diagrams are useful, but must be a supplement to the writing. Now, when you go through this, basically you're explaining the steps of cellular respiration to me. So you would want to be comfortable with glycolysis. If I have no oxygen, I would move into fermentation. You would tell me the two options of fermentation. If you have oxygen, you would do transition prep. You would move into the Krebs cycle and then the electron transport chain. What you would hopefully be doing is just telling me, like, what do I walk away with this step, In glycolysis, I'm going to break, in its simplest of terms. I'm going to start out with glucose, I'm going to do 10 steps, and I'm going to end up with pyruvic acid. And in the process, I will reduce 2 NAD and make 2 ATP. I'm going to do fermentation if I don't have any oxygen, because fermentation allows me to oxidize the NAD so I can continue to do glycolysis. There are two options with fermentation. You could reduce the pyruvic acid into lactic acid, or you could reduce the pyruvic acid into alcohol and CO2. If I have oxygen instead, I will do a transition reaction where I will convert the pyruvic acid into what? Acetyl-CoA. I will reduce 2 NAD along the way and generate 2 carbon dioxide molecules. The Krebs cycle, I will create several reduced NAD and FAD while further oxidizing my acetyl-CoA. All of these NADs and FADs can be brought to an electron transport chain, and we talked extensively about the electron transport chain. It has more hydrogen ions on one side than the other. What's my evidence? I could have a different pH, I could have a different charge. Why do I create that hydrogen ion gradient on one side of the membrane? So I can allow it to go back through and I can make ATP, right? Okay. Now, this one says, describe a lab in which you could measure respiration. Did we do a respiration lab? No. So you are not accountable for that. Okay, so this one, 2B, is off the table. All right, then this will bring us to our last question. Okay, the net balanced equation for photosynthesis is quite simple, but photosynthesis is quite a complex process. So would I expect you once again to be able to tell me the equation for photosynthesis? Yes. Okay, and basically you're going to do the light dependent and the light independent reaction. Then this part, describe a lab in which you can measure photosynthesis, include your setup, control variables, and result. Did we do a lab where we measured photosynthesis?
Yes.
Yes, right at the very beginning. What did we use?
Oh, the leaf disc.
The leaf disc. You hole punched out spinach leaf disc. What did you vary in your experiment?
The concentration.
Concentration of the carbon dioxide you provided it with, because you're going to assume the more carbon dioxide I provide you with, the more product I should get as a result of it. What are the products of photosynthesis that you make?
It's, glucose and oxygen.
Glucose and oxygen. Did we measure in our lab the amount of glucose we made? No. Did we measure in our lab the amount of oxygen? In a roundabout way we did. How did we do that?
We'd see how many floated up to the top.
Yeah, how fast the leaves floated, right? The more discs that floated at a faster time, then those discs, we assumed that solution was better for photosynthesis because we got more O2 generated. More oxygen, decreased the density of the leaf when you had the gases in there and increased the buoyancy, and the leaf would go to the top faster. And remember we made it too, remember where was there ever too much carbon or too much carbon? Yeah, and we found that gave us some false data because we just figured it was so dense when we filled the leaves it couldn't lift itself up due to all that bicarbonate we gave it. Okay? But you could have varied, you could have used the same amount of carbon dioxide. You could have made a 3% solution and varied how much light. And you would assume the closer it was to a light source, the more photosynthesis it could do. You could vary the types of light. You could, what would be like giving it black light? Green light would be like black because it can't use it. So you could try that. You could see if I shine only green light on it, how fast do the discs float? If I shine only white light on it, do the discs float faster? Those are variations you could use. Okay, and then I would remind you, okay, that when we talk about carbon dioxide going, okay, when you say carbon dioxide is going to CH2O, what is that process called?
Reduction.
It is a reduction. Good. What else? What is this process called? Carbon dioxide going to carbohydrate. What is this process called? Photosynthesis. It's easier, right? Don't forget the obvious stuff. Okay, that's called photosynthesis. Okay, when you go from carbohydrate to CO2, what is that process called? Cellular respiration. Okay, now you also told me when you go this direction, you're reducing the carbon, and when you go this way, what are you doing? Oxidized of that carbon. Now when you reduce it, you're having it gain energy, right? So where is the energy coming from?
H2.
No, energy.
Oh, the sun.
The sun, okay? So you're using the sun in order to do this process. Now, you're going to need to provide it with electrons. Where are those electrons coming from?
H2.
Yeah, so that's coming from H2O. You're providing it with those electrons, yes? Now, when we do this experiment here, carbon dioxide to CO2, we're also going to generate energy, right? Because we're doing an oxidation. So what kind of energy are we generating? ATP. Good. And in this one, we actually do what? produce water, right? Because it's the final electron acceptor. Okay, what do these both have in common? They both use what? Yeah, they both use water. What else? What else do they do the same? They both use something. A what?
Oh, it's
A membrane.
Oh.
Yeah, right? It's all about membranes, just like we've been talking about, right? And on that membrane, what do? They concentrate what?
They concentrate the hydrogen ions.
They concentrate the hydrogen ions. What's your evidence that they concentrate those hydrogen ions? The pH is going to be lower, and the charge is going to be what?
Higher.
Yeah, the charge is going to be positive.
Oh, I have a question.
Yes.
So you've mentioned before electrochemical gradient and chemiosmotic gradient. Are those the same thing? Or are
Same
those-
thing.
Okay.
Okay, so why do we do this? Why do we concentrate these hydrogen ions?
So they can make ATP.
Yeah, so when they come back through, okay, we can make it come back through the ATP synthase complex, and you're going to end up generating a whole bunch of ATP. Now, what pushes it across, if you're doing photosynthesis, what concentrates it is PQ, right? PQ concentrates it. If you're doing cellular respiration, and it concentrates it to the inside. Oh, whoops. Concentrates it to the inside. Do you agree with that? But when you're doing cellular respiration, what's doing the concentration of the hydrogen ions? Reductase, cytochrome oxidase. Okay? Okay, you know, your class needs to thank you. You guys did awesome. Okay, and any other questions that you have on that? Okay, good review. Good job, you guys.
Thank you so much.
No problem.